Okay, thanks again. And I'm sorry that you have to listen to me twice, but at least this time I have slides and don't have to ad lib the presentation. So hopefully it will be a little bit more organized. And I will nonetheless try to be a chimpanzee in predicting where things will go in the future. And if I do really good, you can throw me a couple of bananas up here. Gotcha. So I thought that since we were supposed to be looking towards the future here, I would also look towards the past. And I went back to one of the first presentations I ever gave in myeloma, which was exactly 20 years ago. And this is what we were talking about in induction therapy at that time. Induction is the first treatment that you get when you are diagnosed with symptomatic myeloma. This was when I was still at UNC, as you can see in the upper right corner with the logo. And this was a randomized study of VAD, which was a very old regimen with vincristine, doxorubicin, and dexamethasone. And the other version here was with a liposomal form of doxorubicin. So now we think of D as daratumumab and V as bortezomib, but once upon a time, we weren't even dreaming about those drugs. And you can see complete response rate 0, 3%. Boy, that was really horrible back then. So I think the lesson here is that we've really made a lot of progress. I did want to throw up, since UNC won last night, for those of you that participated in March Madness, since I was hearkening back to my UNC days, I did just want to say go Tar Heels for any of you that are North Carolina fans. And now this is what we do for induction therapy, where we've got 3 and 4 drug regimens, but the drugs are much more effective. And we have not just a better overall response rate, but the complete response rate that we now get is often better than the overall response rate used to be. I also thought I would cover the spectrum of things. So this is maintenance therapy 20 years ago, and this slide was showing a study on maintenance prednisone, which is not anybody's favorite thing to do. Steroids, even though they're not considered quote unquote chemotherapy, probably cause more side effects than many of the traditional chemotherapy drugs. And now, of course, we have much better in terms of efficacy and I think in terms of toxicity approaches, including things like lenalidomide. This was what we used to use for relapsed refractory disease 20 years ago, which is thalidomide and dexamethasone. We used to push the thalidomide dose so high that people complained they felt like zombies. This was before zombies were a popular thing on TV shows. And you can see, again, response rates that were relatively modest, but it was the best that we could do at that time. And now we've got so many options that we actually have trouble figuring out which is the best one, which in some ways is a good problem to have. Although, as I'll get to later, we'd like to try to individualize things a little bit better. Overall, what we know is that with all of these novel therapies, there has been a huge improvement in myeloma patient outcomes. And these are SEER data, which look at a number of different hematologic malignancies. Myeloma is sort of on the right side there. What you can see is that there have been one of the greatest improvements in myeloma patient outcomes among all of the hematologic malignancies, although we certainly still have a ways to go. And that's illustrated here. On the one hand, mortality has declined. This is true for Caucasian patients, for African-American patients, for all ethnic groups. But the incidence of myeloma may be creeping up. And we also have more myeloma patients for a good reason, which is that they're living longer. So this continues to be an important healthcare challenge. So I'm going to go through a couple of areas in myeloma and tell you where we are now and maybe what we're trying to do to disrupt that. And then later I'll go into maybe a prediction and ultimately tell you a little bit what you as patients and family members can do. So let's start with smoldering myeloma, which I did earlier. And I mentioned that watch and wait is still often the standard of care. And when we do treat, it's often with the same drugs that are used in symptomatic disease. Does that make any sense? Well, maybe, but on the other hand, as I mentioned, some patients never progress. And since the drugs that we use for symptomatic disease don't cure things there, and smoldering myeloma looks pretty similar at the molecular level to active myeloma, there's no reason to think that maybe the same won't be true there. So as in part I mentioned this morning, one of the things that we're doing is trying to understand the molecular gene profile of myeloma as well as the immune cells. And what this figure shows you is that this box with the C2 on it, those are patients who are progressing from precursor to active myeloma, and the other groups are not. And the good news is there seems to be a very different profile for those patients. So we think that we may be able to better predict who will progress and who will not. And then again, we'll be able to better target therapy. And I mentioned earlier that we also were looking at immune cell changes. So this fancy colored figure shows you that different cells have different properties in different patients. And we think that we're going to be able to maybe block myeloma progression by intervening in different immune cell types. So let's go to newly diagnosed myeloma. One of the things that we've seen these days is we keep adding drugs. It used to be that induction therapy was 2 drugs. Then it became 3 drugs. Now people are talking about 4 drugs. So does that make sense? Well, maybe. On the other hand, one thing we always know is adding more drugs means more side effects. And that's not just in terms of side effects that you feel in your body, but also side effects that you feel in your pocketbook or in your wallet. And we don't know yet whether that will result in, once the disease relapses because you've used more drugs, at that point, it's going to be even nastier and more difficult to manage. So what can we do about that? Well, one thing that we're trying to do is later this summer, we're going to have a trial. And you heard very nicely from Dr. Q about the CAR T cells and bispecifics that look really wonderful in very advanced disease. So what we're going to do is a single agent induction therapy with one of these bispecifics. And we're going to try that first before we give any chemotherapy. The rationale is that if we're getting 70, 80% response rates in people with 5, 6, 7 prior therapies, it should work even better when you give it earlier. And maybe you only need 1 drug instead of 4. It may not work, but on the other hand, if it does, it really revolutionizes the field and lets you get similar, hopefully, outcomes with much fewer drugs. Plus, you save those drugs for later so that if the myeloma does relapse, you still have myeloma that is sensitive to those older drugs. Now, stem cell transplant, you heard a lot from Dr. Q about that. And definitely the dogma is that high dose melphalan is important. And I certainly agree. It's relatively cost effective. The benefit in terms of time in remission and often in terms of survival has been proven over and over again, even with our new therapies. But we do know that these drugs cause DNA damage in myeloma cells, which is good because it kills them. But on the other hand, it can also cause damage in normal cells, which leads to second cancers. And some of the damage in the myeloma cells may lead to mutations that actually help them to survive down the road. So maybe there are better ways of doing this. And although this isn't myeloma data that I'm showing you, one thing that we may be doing in the future are CAR T cells, which you also heard from Dr. Q. And this is actually the ZUMA-7 study, which is large cell lymphoma. One of the leaders of this was actually Jason Weston in our Department of Lymphoma Myeloma here at MD Anderson. And what these curves show you is that the patients who got second line CAR T cell in the yellow curve did better than the patients who got stem cell transplant in the other curve. Now, we don't have these kind of data yet in myeloma. It'll be a few years before we do because these are large studies and the follow-up has to be a long time. But maybe this will be something that puts the autologous transplant out of business. Although Dr. Q, don't worry, he will still have a job because he will do CAR T cell therapy instead of high dose melphalan. So you're safe, Muzaffar, don't worry, I've got your back. Now, maintenance, is maintenance needed? Yeah, right now we do it because it prolongs time in remission and survival. On the other hand, I look at it as a failure because the only reason you need to continue on maintenance therapy is because you haven't killed off the myeloma to begin with. So if you had a better way to kill off more myeloma cells, maybe you wouldn't need to be on maintenance, which many people take for years and years. And although it's certainly much easier to tolerate now than some of the drugs that we used before, like interferon or, again, steroids, nonetheless, it can give you problems which can include second cancers. So in SWOG, which is one of the large groups which gets a bunch of institutions together and we all do the same study, one thing that we're doing is this trial where we are randomizing patients to maintenance either with lenalidomide or daratumumab and lenalidomide. And after 2 years, you get what's called MRD or minimal residual disease testing. That's the most sensitive way that we have to detect myeloma. And if you are negative on that test, then there's a randomization either to continue maintenance or to stop maintenance. And the hope is that people that are MRD negative have so little or maybe no myeloma that maybe they can stop their maintenance therapy, at least for a couple of years. And even if they relapse because of MRD testing, we're going to be able to detect that so early that it will be before they have bone damage or kidney damage. And so we'll be able to jump on the myeloma at an earlier time point. So this is a study that we designed through SWOG, and Dr. Q is actually running here at MD Anderson. So we may be able to challenge this dogma that you have to keep treating, never stop. And hopefully we'll be able to stop for long periods of time, which really makes people happy. And then relapsed refractory disease. Right now we use the same drugs pretty much in the relapsed setting that we did in newly diagnosed patients. And we do that because those are the drugs that we have. But does it make any sense? Because if the drugs didn't work at the beginning, the same drugs or slightly different versions are not going to work better when myeloma relapses. And in fact, we know that the time in remission typically shortens with each subsequent therapy. So what can we do about that? Well, one of the problems is that we use the same drugs for everybody and we don't individualize treatment. We're hopefully soon going to have venetoclax approved in myeloma, which works particularly well in 11-14 translocated patients. So that will be the first drug that we use only in one subtype of myeloma. And then these are just mouse data because I have a lab, so I'm contractually obliged to show you some mouse data. But these are data at a model of deletion 17p, which some of you may know is one of the higher risk abnormalities. And what we have is the mice across the top got just a control treatment. So the myeloma shows up as this red or blue or green color. Then across the bottom, the mice got belantamab, which is an antibody drug conjugate. It's FDA approved for relapsed refractory myeloma. And you can see the mice still have a fair amount of disease. But in that middle panel is a different drug that is also an antibody drug conjugate, but has a different drug attached to it, which works especially well in deletion 17p. And you can see that those mice have a lot less myeloma than the mice below that. And I think this will be the first drug that works better in deletion 17p than in normal, quote unquote, myeloma, and will really help patient outcomes. And by the way, this isn't just a mouse experiment. We have a trial of this drug open right now at MD Anderson, as well as other sites. So I think we're going to be targeting therapy better in the future so that you don't just get combination XYZ that everybody else gets. You're going to get something that is designed especially with the type of myeloma that you have in mind. So I was asked by Jenny to put together this myeloma 2, 5, and 10-year prediction. I didn't know about the monkey analogy that Dr. Q was going to give. But I don't take myself too seriously. So this is what happened when I got my crystal ball out. It said forecast cloudy, check back later. So maybe it's not quite as funny as the monkey analogy, but nonetheless, there we are. Still, I'm going to try to give it my best shot. So what do I think is going to happen in 2 years in myeloma? Well, unfortunately, drug development is a somewhat slow process. So I don't think that in 2 years there will be massive changes. I think smoldering will still be watch and wait. I think we'll still be doing 3 or 4 drugs for initial therapy and transplant, maintenance with 1 or 2 drugs, 3 drugs in early relapse, and then CAR-Ts or bispecifics or other new things in late relapse. When we get to 5 years, then things get a little bit more interesting. I think by then we'll be using some of the triplets in high risk, at least, smoldering myeloma. I think triplets and quads will still be used for symptomatic myeloma. I think if you've got standard risk myeloma, after your frontline therapy, transplant will still be probably the way to go. But I think for high risk patients, CAR T cells may be the way to go. Maintenance will be the same and early relapse. But again, I still think that the BCMA and other CAR T cells will be there. And then where things really get interesting is in 10 years. I think by then for smoldering, we'll be using vaccines or bispecifics as single agents, and also hopefully bispecifics for induction. Now, we may be using one bispecific or a bispecific against 2 different targets, but it will be a purely immune therapy. No more proteasome inhibitors, no more immunomodulatory drugs, no more steroids, none of that stuff. I think transplant versus CAR T if you're MRD positive, but I'm hoping that the upfront therapy will be so good that you won't need any maintenance therapy if you're MRD negative. And I think that early relapse CAR T or bispecifics will be used by then, with some of the current drugs reserved for much later. And so what can you do? And I'm going to sort of paraphrase President Kennedy here and say you should ask not only what myeloma research can do for you, but what you can also do to help with myeloma research. And there's a few things here that you can do. This is, let me go, can we go back one slide? Thank you. There should be one more. Okay, so number one, and you've heard this already, enrollment on clinical trials is really important. The faster we get the answers to these questions, the more rapidly treatment will improve for everybody. And this was a recent study that I pulled out from the JNCI, which looked at enrollment on trials. And essentially what they came up with is that roughly half of patients who were offered trials decided to go ahead and enroll. And half is definitely good, but we could probably improve that a little bit. By the way, the enrollment was similar whether you were Caucasian, African American, Hispanic, or Asian. However, while 50 to 60% sounds good, it turns out that across all cancer patients, only about 1 out of 20 enroll. And the reasons for it being that low are quite a few, including the fact that sometimes there isn't a trial available, sometimes patients aren't presented with a trial, or sometimes the way they're presented with it, it doesn't sound particularly interesting. So I think there's a lot of work to do here to try to improve that. Some of that comes on our end in terms of academia, some of that comes on the industry end, but I think you as patients also need to be advocates for trials, and you should always ask wherever you have a change in treatment needed, is there a trial available for me? The second thing, and Jenny mentioned this earlier, get your care with a specialist. It sounds like an easy thing to say, but there are actually data that back this up. This is from a study that some of my former colleagues at UNC did. Part of that is because trials are more available in places where there are myeloma specialists. But these data actually show that if your myeloma is managed at a comprehensive cancer center, your outcome is better. So here is survival looking at patients who got a consult at a comprehensive cancer center, which is in that orange color, versus the blue color where patients did not. So the question to you is, which curve do you want to be on? Now, you may say, gosh, my doctor locally sees a lot of myeloma patients. He or she is probably just as good as somebody at a comprehensive cancer center. Au contraire, these data show you that even community oncologists who see a lot of patients, and that's in that green curve versus the orange curve in yellow, which is low, there still is a huge difference in outcome between the comprehensive cancer center and the local therapy. And then your next bet is going to be, well, I'll go to the comprehensive cancer center, but it's a pain in the tush to go there. They charge me an arm and a leg for parking, and I'm just going to get their recommendation and get treated by my local doctor, which sounds great. And believe me, I wish that that worked, but these data actually show that that is not the case. Because even when you're on that green curve where you've got a recommendation, but you get your therapy locally, you don't do as well as if you're treated at a comprehensive cancer center. So that doesn't mean everybody needs to be at MD Anderson, but you do need to be with a myeloma expert. That's the bottom line. You heard that earlier, and these are the actual data that prove it. The third thing that you can do is advocate for myeloma research. This is a table from the National Cancer Institute of funding that they have available in different cancer sites. And myeloma is all the way there at the bottom. And it turns out to be 0.9% of the NCI budget. Now, just quick poll, how many people think that that matches the incidence of myeloma? Raise your hand if you think it matches. So the rest of you think it doesn't match? Don't know. Okay, well, I have the data for you. So it turns out that myeloma is about 1% of all cancers. So that's a pretty good match. And based on that, you might say, well, that's fair. If myeloma is 1% of all cancers, it should get 1% of all funding. But if you look at mortality, myeloma accounts for 2% of cancer-related deaths because some of those cancers are curable and myeloma is not yet. So just right there, you have an argument that the NCI should double funding for myeloma because myeloma patients still don't do as well. Now I'm going to get on my little soapbox, not that I haven't been already, but the other thing that you can try to get your folks in Congress to do is to actually do their jobs. So you know that currently Congress is working on a continuing resolution. And that sounds like, it's okay, because after all, they're still in session and you're getting the services that you need. So who really cares if there isn't an approved budget? But in fact, it actually matters. And this is the face sheet of a grant that we submitted last year, which came out at the bottom there at an 11th percentile. That means it was in the top 11% of grants that were reviewed. And last year, the pay line was 11 percentile, so it would have gotten funded. But because there was not an established budget that had been agreed upon by the Democrats and the Republicans, NCI got less money than they were supposed to. They had to cut the pay line to 9 percentile, and this grant was out of the running. Now, fortunately, this year, we resubmitted a revised version that got a 1 percentile. So unless the government goes bankrupt, which, you never know, but nonetheless, it should get funded. But the point is, let's get the money there that Congress intends. Let's have them pass a budget. Let's get myeloma and other cancer research going. And the last area in which you can help. Can we go back by one? Yep, philanthropic support. First of all, because funding for myeloma is not where at least I think it should be, you can definitely help by donating or fundraising, and that can make a big difference for myeloma research. You may think that $10 or $100 or $1,000 doesn't make much of a difference, but it actually can. First of all, the things that are funded by the National Cancer Institute are very advanced. Sometimes we have crazy ideas that can actually make a huge difference, and you don't need a lot of money to generate data to then apply for grants. So even a small amount can make a difference. And there also is greater flexibility in what we can do with those funds. For example, recently we were able to purchase a machine to do the single cell sequencing that I mentioned earlier. That's something that a grant wouldn't pay for because they already expect you to have all of that stuff. And why does that matter? Well, I showed you earlier that grant, which actually, if you looked at the details of it, was for a new drug for T-cell lymphoma. But it turns out that because some of these targets are shared across different cancers, it also works for multiple myeloma. And this is, again, a mouse model where if we can get that box to disappear, maybe we can go back one or two. Thank you. You can see in the box there, what I have highlighted is if you ever hear the term monoclonal protein, that's what it looks like on the serum protein electrophoresis. And that's what gets measured to tell you what your M spike is. And what you can see going across there is that over 1, 2, 3, and 4 weeks after treatment with this new drug, these mice that have myeloma at the beginning develop lower and lower levels. And so we're now at a point where this drug is being turned into something that can be given to humans and moved into clinical trials, probably not this year, but towards the end of next year. And now we do have NCI funding to do this, and this would not have been possible without philanthropic support. So every dollar can make a difference. So let me just summarize, and I hope I haven't gone over by too much, but I think the great news is we've come a long way in myeloma. This is one of the success stories in cancer where outcomes have improved by more than almost any other cancer over this same period. But there's still a long way to go because we probably cure some people, but we definitely want that to be a much larger fraction. And I do think that with some of the tools that you've heard about from Dr. Q and I, and that you'll hear from other speakers later, we're actually at a point where that CURE word could actually be applicable. We just need to do the trials that we can do, hopefully with your support, to show that these therapies work as well as we hope that they do. So thanks very much. This is just the myeloma faculty at MD Anderson, many of which you'll see today. And I'm happy to turn things back over to Greg and hope that I didn't run too far over in my proselytizing up here, or my pontification, I think was the word. Gotcha.